नमस्कार, आप सबका इस खास कार्यक्रम में स्वागत है। आंचलिक विज्ञान केंद्र भोपाल की ओर से हम वर्ल्ड साइंस डे मना रहे हैं और आज ये उसी क्रम में अंतिम प्रोग्राम है जिसमें हमने पैनल परिचर्चा रखी है, पैनल डिस्कशन एंड वी आर गोइंग टू डिस्कस वेरी इंपॉर्टेंट एस्पेक्ट ऑफ नोबेल प्राइज विनिंग वर्क, जो कि थीम भी है हमारे इस प्रोग्राम की, बियॉन्ड द प्राइजेस द इम्पैक्ट ऑफ नोबेल विनिंग रिसर्च। इसमें हमारे साथ एक्सपर्ट्स हैं आईसर भोपाल से एंड वी वुड लाइक टू इन्फॉर्म यू ऑल दैट दिस प्रोग्राम हैज बीन organized in collaboration with Center for Science and Society, Indian Institute of Science Education Research भोपाल, जिसको हम आईसर के नाम से भी जानते हैं। तो इस प्रोग्राम की शुरुआत करने के लिए मैं सबसे पहले हमारे कलीग श्री एनबी साहू जी से कहूंगा कि एक एक करके हमारे आज के गेस्ट का स्वागत करने के लिए हमें बच्चों को मंच पे भेजें। वो बंद होगा, आप बोल सकते हैं।
Good morning, everybody. Now we'll honor our esteemed guests on the dais. I'll invite some children from Carmel Convent Senior Secondary School to please come and offer bouquet of flowers to our esteemed guests. I now request Rabia Ahmad from Carmel Convent School to offer flower bouquet to Dr. Aditya Sharma of Ajar Bhopal. Okay. Thank you, sir, for accepting our bouquet. I now invite Khushi from Carmel Convent School to offer flower bouquet to Professor Satyamurthy, Department of Chemistry, IHR Bhopal. I invite Khushi Kushwaha to come to the dais and offer flower bouquet to Professor Satyamurthy, Department of Chemistry, IHR Bhopal. I now invite Sanskriti, Carmel Convent School, to come with the flower bouquet and to honor Dr. Himanshu Kumar, Physiology and Medicine Department, Ajar Bhopal. Thank you, sir, for accepting our flower bouquet and for being with us on this auspicious occasion of panel discussion.
बहुत बहुत धन्यवाद हमारे तीनों स्टूडेंट्स का, जो कि हमारे इस प्रोग्राम का हिस्सा भी हैं अपने स्कूल और कॉलेजेस से, और मेरे कलीग श्री एनबी साहू जी का भी और पूरी टीम का आरएससी भोपाल का। We would like to inform you that why we are organizing this event. This is the second edition. Previous year we also organized in collaboration with IISER Bhopal only. We thought Nobel winning research work has very important role to play in society, not only for scientific purposes, but also for inspiration and motivation. How a prize or the Nobel winning work can inspire millions of students across the globe to cater their demand and their interests. As a part of that discussion, this came out that we should have curated program considering the audience in the auditorium, and accordingly the program will be designed. We are very much thankful to CS2, IISER Bhopal. They understood the reality and the objective of this concept of RSC Bhopal, and they agreed immediately. And since then, thanks to director IISER Bhopal, he realized previous director also and the current director, sir, and he supported immediately, and we are organizing second edition. We are very honored that we have today three very important, renowned or eminent scientists from IISER Bhopal on behalf of CS2 IISER. Professor Aditya Sharma. Please give round of applause for Professor Aditya Sharma. We have another expert, Professor Himanshu Kumar, and Professor Bharadwaj Satyamurthy. ये तीनों एक्सपर्ट्स अपनी अपनी फील्ड में अपनी अपनी विधाओं में बहुत ही पारंगत हैं, और जैसे कि हम बात करेंगे, आदित्य शर्मा जी एक थ्योरेटिकल फिजिसिस्ट हैं जिन्होंने अपनी पीएचडी कंडेंस मैटर फिजिक्स में यूनिवर्सिटी ऑफ कैलिफोर्निया सांता क्रूज से की है, यूनाइटेड स्टेट्स से किया है, और उसके बाद उनने इंटरनेशनल पोस्ट डॉक्स किया, फिर आईसर भोपाल में 2015 से हैं, और उनका रिसर्च जो है एनालिटिकल न्यूमेरिकल मेथड्स और एक्सप्लोर करता है कनेक्शन बिटवीन स्टैटिस्टिकल फिजिक्स एंड क्वांटम इंफॉर्मेशन। जिसको क्वांटम मैकेनिक्स में इंटरेस्ट है, आगे चल के क्वांटम कंप्यूटिंग में इंटरेस्ट है, क्वांटम वर्ल्ड में, तो क्वांटम इंफॉर्मेशन इज वेरी क्रिटिकल एंड इंपॉर्टेंट सब्जेक्ट एन एरिया ऑल्सो। And we have Professor Kumar, Himanshu Kumarji. He is here. He will talk about related to his work in immunology, and especially he's going to represent from biological sciences. He's a renowned immunologist. He specializes in innate immunology. His research focuses on regulations of antiviral immune responses, particularly during flu, dengue, and other viral infections. He earned his PhD from Osaka University and has been a faculty member of IISER Bhopal since 2010. He is recipient of numerous national and international awards, and very prestigiously, we are very honored to have him on this August gathering. He is also editor-in-chief of International Reviews of Immunology. Please, once again, to both the experts. And now we have Professor Bharadwaj Satyamurthy. He's a renowned chemist. Chemistry जिनकी subject है, you can relate to him. The world is very much important because without material and interaction and chemical reactions, we are, I think, nothing in this known cosmos. And he completed his BSc and MSc from University of Madras, and afterwards his higher education from IIT Madras as well. He obtained PhD from SUNY Buffalo, again from United States American University, and he earned under the guidance of very renowned scientist, Professor Thomas, how to pronounce it? Sapasti. Sorry, Professor, if I'm not pronouncing correct the name, but definitely your work has inspired millions across the globe. And he also finished his postdoctoral research from the University of Michigan and Duke University. He joined IISER Bhopal, I think both in Professor Sarma and Professor Bharadwaj in the same year. So once again, for Professor Satyamurthy. So जिसके लिए हम सबका इंतजार है कि आज हम इस प्राइज को लेके खास क्या बातें करने वाले हैं. So this program is designed in a manner which we will have 3 rounds. पहले एक छोटा सा कि नोबेल प्राइज जो फिजिक्स में मिला है वो आखिर क्यों, किस फील्ड को मिला है, कौन उसके लिए जो साइंटिस्ट हैं उनके उस काम को नोबेल विनिंग कमिटी, नोबेल कमिटी जिसे कहते हैं, रॉयल सोसाइटी ऑफ स्टॉकहोम जो है रॉयल सोसाइटी, उनके द्वारा जो दिया जाता है, तो वो क्या कंट्रीब्यूशन है, उसके बाद हम जानेंगे केमिस्ट्री के बारे में, उसके बाद हम जानेंगे फिजियोलॉजी एंड मेडिसिन मींस बायोलॉजिकल साइंस के पर्सपेक्टिव से. तो वन बाय वन आई विल रिक्वेस्ट ईच प्रोफेसर कि वो अपनी बात को संक्षेप में 5 से 7 मिनट में वो जो हमारा एक पावर पॉइंट प्रेजेंटेशन के माध्यम से एक एक करके वो आपके सामने रखेंगे. देन वी विल हैव अ कॉमन डिस्कशन in interaction, and afterwards we'll open this floor for question and answers. If any question comes from our YouTube live, so I'll ask my team कि वो हमें बताएंगे, तो हम वो सवाल भी शायद अंत में प्रयास करेंगे कि जोड़ेंगे. और we will talk in bilingual format, mostly हिंदी भी मिक्स रहने की कोशिश रहेगी, ताकि जो ऑडियंस हमारी केंद्रीय विद्यालय के अलावा जो दूर दराज के स्कूलों से हमें देख पा रही होगी, उनको भी हम जुड़ने का एक मौका मिलेगा. इसी बात पे एक बार जोरदार तालियों के साथ हमारे आज के स्पीकर्स, thanks to IISER Bhopal, on behalf of our council, National Council of Science Museums, let's start this program. And on this note, I'll request Professor Sarma, ki please, sir, let's start the program. And the mic is in your hand. You can continue.
Okay. Good morning, everyone. Okay, so it's a great pleasure to be here and to see so many really young faces, lots of excitement. So my goal will be I'm given about 10 minutes. I have a few slides prepared to briefly describe what the Physics Nobel Prize was about, who the scientists involved were, and what were their contributions and why it is exciting and how it can lay the foundation for exciting times ahead. Okay, everyone can hear me? Okay, okay. Um, is it working? Okay, so this is a slide which is basically a taken from the official Physics Nobel press release. So the two people involved this time, so who got the Nobel Prize in Physics are John Hopfield and Jeffrey Hinton, and so both of these, gentlemen Okay, is this good? Okay. Yeah, so they have worked in a variety of fields. So it's in the true spirit of interdisciplinary research. So Jeffrey Hinton, for example, has made lots of contributions in the field of computation. Although the ideas which go into some of his innovations are rooted in physics, like I will try to argue. And John Hopfield has straddled across a multiple disciplines, biophysics, computation, and a variety of fields. And so the key contribution which led to the Nobel Prize for Hopfield was this so-called Hopfield network, which is given which is a way to come up with a new innovative way of um, storing memory, right? So associative memory, and Jeffrey Hinton built on these ideas and um, made a more sort of generalized version of the Hopfield network, and goes by the name of the Boltzmann machine. So, like the citation itself says, so for foundational discoveries and inventions that enable machine learning, as I think everybody in this room has, have you heard of the word machine learning? Right. So, learning and artificial neural networks. So they're very closely related ideas. But the key ideas which went into the current explosion of ideas that we are seeing today were spawned by these two gentlemen. Right. So there's a little bit of a history of where they were born, and so they spent a lot of time in Chicago. John Hopfield, and I think Hinton spent most of his career in the University of Toronto, and so the ideas are sort of described here. So basically the idea is that John Hopfield found inspiration in the human brain. So maybe to explain this, I will ask you a question. So did anybody have an aloo paratha this morning? Okay, aloo paratha there. So only one person, nobody else had how was it? Did you have a really happy breakfast? Okay. Yeah. So the reason I ask is, have you ever experienced this phenomenon where suppose you entered a room and maybe your mother has been cooking aloo paratha that day, and the moment you entered the room, you know that that's what's been cooking there. The smell has already told you this. Has anybody had this experience? So the moment you have smelled something, your brain already is aware that there is a connection that the brain has made to some past experiences, right? So a question which can come up in your mind is, how is the brain so quick to make this connection between an experience that you're experiencing today to something that is stored in the brain long ago, right? So you might wonder how how memory works in general. And so in fact, we are all made up of like zillions of experiences. They're all stored up in the brain, and if the brain has to actually connect between any current experience with all this, let's say that there's a huge register sitting inside the brain, and if the brain has to go to the register and open every page and go line by line and ask, okay, does this connect to this experience, does it connect to this experience, and so on. If it were to do in this kind of a sequential order, it would take it a zillion years perhaps to figure out this point. But that's not how the brain is working. Immediately, as soon as you entered the room, you know that your brain has connected to aloo paratha that you have eaten in the past, and perhaps some other images where you had maybe your favorite restaurant. So many things which are associated with that experience, current day experience, are immediately connected to something in the past. So how does the brain manage to store memory? It's very different from the kind of memory that we store in the form of random access memory in a computer, for example. Right. So it was these kinds of ideas which John Hopfield was pondering, and so there were already models within neuroscience which said that what the brain does is actually to associate ideas with, there's a kind of a descent which happens, a descent in energy, which I will show you in the form of okay. So before I show you a picture, I'll quickly tell you what the current day methods for building a neural network are. So in a traditional computer, what you would do is you have a set of rules, right? I mean, how many of you have done programming here? Anybody has written a program? So when you write a program, you tell a computer, you take a step one, then you do step two, step three, so on. All the steps have to be written down, right? If you give detailed instructions, a computer is very smart and is able to do it fast, right? That's what a computer is capable of. But the radical new idea here is that you basically get your computer itself to design the steps that it's supposed to do. And how does it do? It's very closely related to the way your brain managed to quickly connect back to some past experiences, right? So in machine learning, the computer is fed lots of examples, and it takes all these examples and actually learns it, manages to in some sense write its own program, right? And the inspiration for the so-called artificial neural network, it actually comes from the human brain. The brain has lots of neurons, and these neurons are mimicked in this kind of a model by nodes, and then these nodes are in there are interactions between nodes which are to be set in the process of something called training. um, yeah, so this is a schematic of neurons, actual neurons. So connections between neurons are weaker, right? I mean, suppose you had aloo paratha, and then the restaurant in which you had probably like red color background, let's say. So maybe there's a connection between red and aloo paratha, right? So these two things which are apparently completely unrelated may have happened together, and your brain would have made these connections. And then maybe there's something else like which is going on the same day, and that may be a different memory in your head, and those things are not connected at all. And so a crude way of setting this up in a computer is to think of these neurons as just these nodes, and each of these nodes, for example, can either take the value 0 or 1, and then there are these bonds, right? I mean, within statistical physics, one thinks of there are models for magnets which which are very closely related to these kinds of models. And so Hopfield realized that we can use write down an equation like this. We can think of neuron a network as a kind of the states that your neurons can be in together, they form a network, and then you can associate a certain energy with it. And then uh, you know what what your setup would do is it would keep on updating these spins or neurons. I mean, some of them can be 0 or 1, right? I mean, you keep on changing them in such a way that your energy keeps on decreasing. And so this process of going downhill, there is an algorithm which one can write down, right? I mean, it's a matter of fixing these interactions, right? These interactions and so-called biases are in the field of statistical mechanics, we call them magnetic fields. And so there is a way for your system to keep on rearranging the states of these neurons so that the energy will keep on going down, and then you will reach one of these basins, right? And so this basin is where your memory is stored. I mean, this memory, that is the reason why you're able to very quickly go down, as opposed to a traditional form of storing memory where you have to do sequentially. Here you just have to sort of descend down in energy with a small set of steps. This is what roughly this is what the brain does when it's able to fetch memories. And so this is the algorithm. I mean, the technical language, you can look up and read about it. But so this is a very nice picture which is again taken from the Nobel website. So basically, the idea is like, I mean, you can think of your memories as part of some huge landscape. So there are hills and valleys, and so when you drop a ball from uh, from some random position, so you're depending upon where it drops, there is a tendency for it to sort of spontaneously slide downhill, and it gets trapped in one of many possible valleys, right? I mean, it's what are called metastable states. Wherever it ends up landing, that corresponds to a particular memory. So for example, if you want to store certain patterns, like for example, I want to store a pattern like J, the letter J, and maybe this letter J corresponds to a particular metastable valley. And if I were to give it something which looks like J but not exactly J, your system is able to sort of spontaneously find its way, and then it's going to remember a memory which it has been tuned with. Similarly, you will have some other part of your landscape which corresponds to the letter I, and so on. So there is a quick way for it to uh, recover this memory. And so this is the idea of Hopfield. And then came along Hinton, who said that, okay, why don't we do it also introduce the concept of temperature, right? So I don't know, some of you would have seen that magnets, if you take a magnet which can lift iron and things like that, but if you were to cool it below a certain temperature, if you heat it beyond a certain temperature, the magnetism is gone, right? Is anybody aware of this? So yeah, so this is called a ferromagnet to paramagnet phase transition, right? And similarly, there are more fancy magnets which are called spin glasses, which have order where in a ferromagnet all these local magnetic moments tend to order, right? You can think of these as spins which can be either up or down, very similar to these neurons which we said could be take either value 0 or 1. And so these spin glasses where you have more complicated kind of ordering, which also happens at a temperature below a certain critical temperature, right? So the idea was whether, in fact, this Hopfield model was very similar to a very famous model in spin classes called Sherrington-Kirkpatrick model. And so Hinton said, let us also include a temperature in this. So now there is a competition. There are two different kinds of forces which are in play. If you have seen in a high school study of thermodynamics, there is this notion of entropy. So systems have a tendency to increase entropy while also tending to decrease energy. When both of these forces are in play, the role of temperature also is important. And so then you have your system will descend down. So now you have a probabilistic description one has to put in. So you still have the same kind of energy, but it's not necessary that your system is continuously falling in energy. Depending upon the temperature, there will be certain moves where it may also go up and then come down and so on, depending upon the particular temperature in play. And so this eventually gives you a stochastic aspect to this. And Boltzmann is a little more general. Boltzmann machine is a little more general. And in particular, what it can do is it can actually give you, it can generate new kinds of pictures. For example, if you were to feed 1,000 pictures of cats to a model like this, and you train this model to say, okay, this is a cat, this is a cat, this is a cat, and all these bonds get arranged according to what cats are, and then you can ask this machine to generate a new picture of a cat, which it can do. So this is what is called generative AI. I don't know if any of you has played with modern AI tools where it say you can say, give me a picture of a sunrise, and it can give you a picture. Has anybody tried this? How is it doing? So underneath this is, some very sophisticated version of the Boltzmann machine that Hinton came up with. And so then there's a more advanced version of it. It's called the restricted Boltzmann machine, where there are hidden layers, and then you have all these ideas of deep learning and so on, where basically you have, these units with which you connect to the outside world, but then there are all these hidden units which you can add. And so in fact, the kind of AI tools that you can, it's available with Google and ChatGPT. How many of you have played with ChatGPT? So ChatGPT will use like lots and lots and lots of layers. So these are called deep uh, deep learning methods, right? I mean, so there are sophisticated algorithms, and so but ultimately, so the base for all of these ideas is the Hopfield network, which is like here, and then the Boltzmann machine, where you have visible and hidden layers. Okay, so yeah, I think now it's better. So you have Boltzmann machine where you have the idea of a visible node and a hidden node, and a restricted Boltzmann machine is one where you have visible and hidden nodes, but the connections are restricted in a special way. So there can be no bonds between visible nodes, whereas visible nodes are only directly connected to hidden nodes and so on. So this is a technicality which we don't want to get into here, but I'm just saying that this dramatically increases the capacity of this kind of a machine to store memory and also to generate new possibilities. So you can have in the future, you can imagine machines like this generating new kinds of music. So you train it with you throw 1,000 songs in a particular rag, for example, and then you ask it, can you generate a new piece of music in the same rag, and it's possible for it to do. And the reason is because there is these kinds of fancy networks which are in play underneath. Um, is this working? Yeah, okay. Yeah, so I want to end my talk, but before that, I just want to quickly show you a poem that I got ChatGPT to generate for us. So, in the depths of data where patterns reside, neural networks awaken their power to guide. Layers upon layers, they weave and they learn with weights that shift to discern and return. From images to words, they see and they hear, transforming raw inputs, removing the fear of chaos, confusion of things undefined. They find the connections that once were confined. But deeper still, in the shadows they wait. Boltzmann machines rise with a stochastic state, where states are entwined in a dance of descent. They seek hidden truths with probabilities bent. Each node a flicker, a spark in the dark, engaged in a random yet purposeful arc. A symphony forms the noise and the heat. A model emerges both simple and neat. In each wait, a potential is found emerging from chaos, a structure unbound. Through Gibbs sampling's rhythm, they settle and blend, a probabilistic dance that will never quite end. The network remembers, learns, and adapts from the shifts of the weights to the quietest labs, a fusion of logic, of art, of data, a neural connection, and evolving heart. So it's unimaginable that a machine could have done something like this. Maybe even 10 years ago, if somebody had told me that it would be possible, it would have been hard to believe. But all this is happening with there is some underlying neural network which has basically figured out connections and written a very advanced poem of this kind. Okay, so on this note, I will say thank you.
Please, please. Thank you for your very wonderful explanation about this year's Nobel Prize in Physics and the very core idea why this work is Nobel winning and how it affected the entire, primitive years of, I should say, machine learning, which actually changed the course of generative AI also. So thank you once again. Please let us give a big round of applause for our Dr. Sharma. And now, considering the time which we have at our hand, we'll immediately request Professor Bharadwaj, please, sir, take this seat and we'll hear him about this year's Nobel Prize in Chemistry.
I hope I'm audible to everyone here. Yeah. So once it goes to full screen, let me get started. Before that, let me thank Regional Science Center for organizing such an important event, especially for school kids, and making sure the schools also encourage students and also from first year students to actually attend an event like this. This hopefully would be very helpful given that we have wonderful speakers from IISER Bhopal representing their own branches of science. So, okay, so here starts my presentation. So I'll be only discussing about the chemistry Nobel, and I'm sure many of you would have seen this is kind of the medal that one ends up getting when they win the Nobel Prize. Okay, so where should I change the slide? Yeah. So generally Nobel Prizes are awarded to 1 to 3 people in general, and they are shared. As Aditya also showed, it's a 50-50 share between 2 people in physics. In chemistry, it's a share between 3 people, where one person gets 50% by name David Baker, who's from University of Washington, and he got the Nobel Prize for computational protein design, which we'll try to understand what it means piecewise as we go. The other 50% was awarded, just a moment. Is it a little slow to respond? Can you go back? I'm going back. Um, is there a pointer control there? Can you please go back? Yeah, that'll be better. So I'll be telling next for each slide. So then remaining 50% was given for protein structure prediction to Hassabis and Jumper, who actually are part of Google DeepMind. So in this aspect, I thank Aditya to have given a wonderful introduction of how artificial intelligence and machine learning are useful towards applying in different sciences. Of course, he tried to show the basics of how they work. Here, we'll be trying to see an application of artificial intelligence that helped an important problem. Okay, as we go forward, what is common to both the contributions? So one is for computational protein design, and the other is for protein structure prediction. So you guys see a common word between these two? Yeah, of course. Go forward, please. So now let's try to understand. We understand the word protein is common between those. I'm not sure how many of you know what's a protein. So I'll try to emphasize on why it is important to study proteins and how do they affect and how do they help us towards society, and try to slowly understand the other terms such as computational, design, and structure and prediction. So let's move slowly piecewise. Okay, so what's a protein? Proteins are essential molecules in our body. I'm sure you guys are studying that in science. And they are major part of structural components of body tissues like muscle, hair, your skin. And they also act as catalysts in order to make sure your reactions happen in the body, right? Which is also why at times you get fever, whereas if some of the enzymes are not able to perform, the temperature is increased a little bit so as to make sure they still catalyze essential reactions so that you are still alive, right? So therefore enzymes are important. And antibodies, things that fight diseases. My colleague will speak more about it as we go forward. But how do we get proteins? Mostly from the food we eat, and the food that contains them are eggs, cheese, paneer, nuts, peas, milk, fish, and red meat, and so on and so forth. So that's how we get most of our proteins from, and we do synthesize some of them ourselves, meaning that amino acids. So what happens if we do not have enough protein? So this is an important question, isn't it? Okay, why is protein important? If you see here, if you have a protein deficiency, one ends up having malnutrition and ends up having problems like marasmus and kwashiorkor. Okay, so this being the case, we all understand proteins are important part of our body, and therefore we have to understand how they look and how they behave so as to make sure we benefit from them. So what are protein molecules really? Protein molecules, I'm just trying to show an example of a water molecule here. All of you would know water is composed of H2O, correct? So what I'm trying to show here is that the red one is the oxygen atom, the two white balls are the hydrogen atom. Okay, so from here on, you're going to see a lot of structural models of proteins. And what are proteins made of? They are made of something called amino acids. Amino acids are characterized by a 4-carbon center, which is attached to a carboxylic group, an amino group, a side chain, and a hydrogen atom. If you do not understand this, do not worry. We'll play with it like building blocks so that you understand how they end up coming. So if you go back, please, one at a time. So if you see here, the side chain group varies across 20 different combinations, which are the amino acids that are present in nature, out of which 7 to 9 are essential, meaning that you have to eat in order to get them. You cannot synthesize in your body yourself. But interestingly, you can have any such combination. When the amino group reacts with the carboxylic group, you start forming bonds, and then therefore you start to have a polypeptide, meaning that multiple amino acids put next to each other form a polypeptide. So when you have such polypeptide in a given sequence, it's called the primary structure. If you're wondering what we are talking about here, if you understand alphabets, if you put alphabets together, you get a word, correct? And now the word also assumes a form which has a meaning. So basically, the words put together could have a certain meaning associated with it. In this case, when the amino acids are put next to each other, they end up assuming a certain three-dimensional structure. Like your fingers are of a certain kind. Your fingers are not proteins, but I'm just giving you an idea. Okay, different body parts have a specific structure associated with them, which will come as we go forward. Multiple of such secondary structures get together to form something called a tertiary structure. For instance, while your hands look like this, everything put together looks like a body. Each of the features of the body parts are quite different, and when they put together, you have a certain unique human being with a certain height, certain weight, and certain characteristics. Similar are proteins. When you have different sequences that you put together, you get a secondary structure and therefore a tertiary structure. And then when multiple of such tertiary structures go together, you have something called a quaternary structure. It's something like I and Himanshu and Aditya form a quaternary group to come here to discuss with you, right? So this is how proteins also work. You have a string of a sequence, and therefore you are able to go from the sequence to something like a complicated structure. So now let's take some examples to understand what are proteins. I'm sure we are breathing. Are we? We better be, right? So can you please click on this video? Go back, please. Please click on the video on the left. So this is a representation of the, go back and there'll be a play button at the bottom. If you hover your mouse over this. So basically, this is a protein molecule, hemoglobin, that allows us to breathe. And this is the complex arrangement of atoms. I showed you H2O, but now this is composed of a lot more, thousands of atoms put together. And it has a specific structure. And if you see, these are the heme groups that allow you to transport the molecular oxygen from outside to inside and take the carbon dioxide outside from your body to, I mean, from the inside your body to outside. Makes sense? So therefore, the structure of this protein matters a lot in order to do the function of transport. This is another protein. This is a potassium pump protein. We all have cells, and our cells interact. Like Aditya was trying to say how neurons interact. They generally tend to interact with ion exchanges. And this is one such protein which ends up moving potassium ions across the cell membrane. And this is the three-dimensional structure of it. So basically, proteins' shape are very important for their function, right? So where can all you guys take a look at these structural models? Go search in Google called Protein Data Bank. You'll be able to find the link here, and you'll be able to find the specific protein structure, and you'll also be able to play with the 3D model. So you can have fun rotating and taking a look at how these things look. Okay, now quickly functions of protein. I'm sure you already are able to understand it performs many essential functions like the cellular process of replication, transcription, translation, cell division, cell structure, as I said, hair, skin. They are also enzymes, meaning that they end up catalyzing very critical reactions, signaling, transport, and so many other processes. Okay, fine. Structure determines function. What does it mean? I'm sure you guys are all using keys for different purposes, and you know for a fact a certain key that opens your bike or car will not open your home, and vice versa. And I'm sure you have different keys for different purposes of your certain doors, homes, offices, and so on and so forth. Similar are proteins. Their structure ends up driving their overall function, and based on the function, it assumes a certain structure to perform the same. Right? I hope that makes sense to everybody. Okay, now what is really the problem in understanding protein folding? I said there are 20 amino acids. The number of sequences could be 50 to 100. Like the alphabets that you put together could be 50 to 1000 actually. The largest protein is about 3500 amino acids. Now, if each of the amino acid could sample 3 states, which I'll try to show later, an alpha helix, a beta sheet, or a loop. Now the problem is, if you have 100 amino acids, it can possibly sample 3 power 100 states, which is about 10 power 47 states. There are way too many possibilities for it. Even if you assume the protein is going to try each of that for 1 picosecond is 10 power minus 12 second. It's going to take about 10 power 27 years to go from a single strand to a folded structure. So it was apparently seen much earlier on in 60s and 70s. That's not going to happen. The protein is going to search for every possible conformation before it attains its overall fold. Okay, so this is where we stand on the shoulders of giants. It's not always a Nobel Prize is given only for that year's contribution. Significant contribution is done by people much in advance. So these are two major contributions from Anfinsen and Levinthal, where Anfinsen was able to show how a certain type of bonds actually predetermine a protein to fold. And Levinthal was the one who gave the paradox. If you're having 3 possibilities and 100 amino acids, it's never going to look for every possibility before it folds. Therefore, the primary sequence, that is the sequence of amino acid, already decides a tertiary fold. Yeah, that makes sense. So therefore, structure determines function. So for proteins, you have a primary structure, which is sequence of amino acids that decides, for instance, this is the sequence for hemoglobin that determines the secondary structure, which you have either alpha helix or a beta sheet. And when these are put together, you have something like a tertiary fold, and many of these tertiary folds go together to form a quaternary structure. Does that make sense? So that's a protein, and that's how protein assumes a fold, and the fold of the protein matters for its function. Are we in page? Yes? Next, please. So proteins are of very different kinds. There are 200,000 protein structures that have been solved thus far in the last 50 years. The first structure was myoglobin. And depending upon the structure, functions will be performed. That's the hemoglobin I was talking about. This is the chromatin DNA, where the DNA gets wound in each one of our cells. The DNA in our cells are about 1 meter. If we take literally each DNA from our cell, we can go to the moon and come back. It's that much, but it gets wound within 1 micrometer, much more smaller than the size of the cell itself, right? So there are different proteins that help in folding, that help in interacting with DNA, that end up having proteins being prepared from DNA, and so on and so forth. So proteins matter, their fold matters. Understanding how they fold helps us understand biology, right? Forward, please. So fun fact, if you guys want to go and play with it, until a point that people were able to go from sequence to structure, a video game was developed by the name Fold It. The website is right there, where students could actually go and play and help a protein fold and figure out what happens. To your amazement, there were many protein biophysicists who couldn't understand, but many kids like you were able to play this video game and actually find out how a protein folds. So go check this project called the Folded Project. You will learn more about it. Next. So what you see here is a sequence drive structure. But if given a sequence, it's not certain whether one can predict the structure, right? For instance, next please. So for instance, a single mutation could completely change the protein structure. So this is confusing. It's something like the word PUT is put, but BUT is but, right? You end up using the UT pronunciation based on which alphabet is in front. So similarly, in a protein sequence, just by changing a single amino acid, it could completely destroy the structure. As an example, you would see for sickle cell anemia, where the hemoglobin structure changes drastically. On the other hand, there are also other examples where you change 80% of the sequence, there's literally no change in the structure. So this is very confusing. As he was rightly trying, Aditya was trying to point out, when you have some memory, when you want to invoke it, then you have to make sure other parts of the memory are properly invoked. Otherwise, you're not able to relate to what you wanted to do. So this is the confounding protein folding problem, where a single change could completely destroy it, or a large change doesn't do anything to it. So this is where the integral problem of protein folding had to be solved. Okay, so when do we say we understand something? When I ask you whether you know addition, there are two ways of answering this question. One is that when I say what is 5 plus 3 and I say it's 8, you guys say yes, it's 8. Yeah, that's correct, right? That's one way of understanding, meaning that you validate a result that's an observed outcome. You know what it is, and you're able to say, yes, that is the actual outcome. On the other hand, you could also start predicting. If you really understand addition, if I say what is 34 plus 63, you'd be able to say what it is, right? So these two are the way scientists test themselves whether they understand something. So the whole thing that the Nobel Prize is based on is to understand, when I give a sequence, can I give a tertiary structure, meaning that an overall fold. Okay, now let's take a look at the first part of the Nobel Prize, which is the protein design part, which is won by Professor David Baker from University of Washington. Forward, please. So this problem has existed for a while. So people wanted to ask, let us say I have power over protein folding. That means if I have a certain design, I should be able to come up with a sequence for it to fold in a certain way. What am I trying to say? Not all the words, you know the spelling. Some of the words, when I say, you make up the spelling based on how it sounds. So they were trying to say, if I have a certain fold, can I make a computer come up and tell me a sequence that does not exist in nature to have the same fold? So this is one example of a protein called a zinc finger protein, meaning that a zinc atom is bonded to a protein based on some histidine and glutamate interactions. So the question was, can we fold it without the zinc? So there are many attempts made where people were able to come up with about 33% identity, meaning that they changed about 67% of the protein. They were able to get a fold. But interestingly, in the later part of 90s, what one was able to do is to completely change the sequence, still have a similar fold, but without the zinc ion, which matters a lot for such folds to exist. Just 2 minutes. So keep going forward. All right, so therefore, people wanted to come up with protocols where if you have a certain backbone, can I come up with a sequence? Next, please. So that is where David Baker's contribution comes through. Back in 2003, he was able to come up with a far much more complicated fold of the protein, which has a mix of alpha helices and beta sheets. And he was able to come up with a computer-based program called Rosetta, where he gives a certain fold. He gives this kind of a fold, alpha, beta fold, and therefore says that, computer, give me a sequence that does not exist in nature. And they were able to do it, and they were able to show literally the fold they wanted, the sequence actually does. Next slide, please. So that's where his major contribution is. So the computational part comes from the Rosetta program, meaning that all the structures that exist in PDB, they were able to learn from that, and using that, they were able to give a certain structure, give us, and therefore predict a sequence for it. Next slide, please. So this is what is called de novo design. The word de novo stands for Latin and from the beginning. And of course, one has to optimize structures and then obtain a given structure. Simulations were done to stabilize. So basically, what did David Baker designing module contribute towards? They were able to design proteins, basically de novo enzymes that could catalyze things that are not naturally possible, and also come up with high affinity and selective binding of small molecules. We'll see more of it as we go in the next slide. For instance, next slide, please. So you're able to see he was able to computationally design a nanoparticle which is just made of proteins. This is a huge protein by itself. At the same time, he was also able to come up with a protein that binds a molecule called fentanyl. Okay, and therefore they were able to sense these molecules as sensors, or also make molecular motors and also other geometric shapes that are useful for many purposes, including drug discovery. Okay, now the protein design part is done. Let's ask, what is the protein structure prediction? I already told you protein structure matters for function, but we do not know when we start with the sequence what will be the structure. And what these gentlemen, that is Hassabis and Jumper, were able to do from Google DeepMind, which is a company from Google, where they're trying to use AI-based algorithms, like Aditya had mentioned, to solve the hardest scientific and engineering problems. And one of the major outcomes that has come from AI towards a real life solution is actually the protein folding problem. So interestingly, this also has a long past. People realized after Anfinsen and Leventhal, we should be able to predict things from sequence. So what people used to do is to take protein sequences solve their structures, and make the computation people compete. For instance, I give you a mathematical problem. I know the answer. I wait for you until you find the correct answer. If you got the correct answer, that means you understood what is the problem and how to solve it. So this is what people were doing. People took different sequences, solved their structures for it, and in parallel, people used to predict without any information what should be the fold. That project is called the critical assessment of protein structure prediction. Interestingly, when I did my PhD, I had also contributed towards 2 of such millions of structures towards such a prediction. So interestingly, these were all more brute force, and there were some molecular dynamic simulations that could be available. But interestingly, in 90s, with a lot of DNA sequencing, very good advancements in biology, DNA sequencing and recombinant technology, people were able to make different types of proteins, and techniques such as X-ray crystallography and nuclear magnetic resonance, which I work on, were able to cater towards solving these structures at a much larger throughput. So therefore, what happened is the database improved. But interestingly, in 96 itself, they were able to realize if we understand the contacts of the atoms, we'd be able to figure out what's going on. This is the example like Aditya gave. If you have an information of J should be embedded, if you give something close to it, can it pick it up? That's basically that application. And that started happening back in 2016, once again by Hassabis and other group. Next slide, please. A few more clicks, please. Yeah. So in 2018, Hassabis was able to use an AI-based algorithm called the convolution neural network to actually predict two-dimensional distance map between atoms. Okay, that program is called the AlphaFold. This was the first version of it. While it was a good leap, meaning that this is the level at which you can predict, and you're able to see in 2016 to 18, they were able to predict much better. Let's say 40% is the pass mark in an exam you have, and these guys were able to go close to 60%. But the interesting aspect is that as they furthered things with Jumper, they were able to come up with even better algorithms and improve the AlphaFold prediction very close to 90%, which says if you give a sequence, one can actually predict the protein structure just using a computer without any experimental data, giving an accurate and precise information. So with that being the case, where are the applications for it? Especially during the pandemic, the SARS-CoV-2 protein was modeled using these techniques, which are useful for finding drugs. For instance, when a biologist ends up finding a drug or a drug chemist finds a drug, they have to understand the protein structure to figure out where the drug will bind, therefore to cure the disease. And there are many other components of it. The most important aspect that came last night is that the third version of AlphaFold, which was released before the Nobel Prize was won by these two people, is that now it's open source. What does open source mean? Literally, you can install it in your computer, give a certain sequence of protein, and see how it might fold. So that's where science goes in terms of contribution towards a society. And this way, people could take very different protein structures for different diseases and try to solve, okay, what is the structure for a given protein? Can we find drugs for proteins that are not easily solvable in terms of structure? So with that being the case, what is my association with it? Sorry, a little bit of steal of glory here. I happen to work with the person who found the first top 7 design protein by name Brian Kuhlman, who worked with David Baker. And I had the opportunity to solve a de novo structure by myself when I was doing my PhD. And currently, what do I work on? So it's actually a 4-helix bundle. They predicted something with NMR spectroscopy. I was able to see whether it's close to their prediction. And in addition, currently what I'm doing is looking at DNA junctions called telomeres, which are very important for many biological processes as they are found in many different regions. I'm asking the same biophysical question. If you give me a sequence, can I predict its structure? And why is this important? It is quite involved in many diseases, including cancer and neurodegenerative diseases. With that being the case, where do we stand to finish up? While designing sequences to a precise fold has been achieved by Baker, and structure prediction from sequence has been achieved by DeepMind people, Hassabis and Jumper, the question still remains open. Why does it happen? For instance, you got the right answer, but you do not know how you reached the right answer. So that is still an open question in terms of science that is available for people like you and me probably to contribute towards it in the next coming decades. And also there is a twist to the story. Not all sequences go to a certain protein. There are examples where a certain sequence will never fold to something proper. These are called intrinsically disordered proteins. And my biology colleague will take it up in terms of the microRNAs, which are also kind of similar to the IDPs, which have very important function in biology. And before I finish, I'm not a very big poem person, but I'm very happy that Aditya showed something from ChatGPT. This is from a very well-renowned poet by name Robert Frost. I'm sure you guys would have heard. I'm going to skip all of this, and I'm just going to say the proteins are lovely, dark and deep. It's not that easy to find what they are. Of course, I switched the word from woods to protein, but there are many promises to keep in terms of us solving problems of what proteins are and how do they contribute towards diseases. And there's miles go before we sleep, and there's miles to go before we sleep. So remember this and keep pushing to understand how biochemistry works. Thank you.
So thank you, Professor Satyamurthy. And I'm really glad that the way he explained very beautifully about the concept and the Nobel winning work of those three gentlemen. One is dedicated for the science designing a protein, which was not a part of our earlier in our universe, I should say safely, because the research is clearly mentioning that is created first time. But now how machine can help. So whoever is studying artificial intelligence, machine learning, deep learning, try to understand these are not only core areas of technology where you will only excel in those certain areas. You can apply and use in the field of sciences for advancements. So without further ado, because considering the time challenge now we have at our hand, I'll request Professor Kumar, please take your seat, sir. Thank you.
Yeah, we are close to afternoon, but yeah, good morning right now. and so first, I would like to thank Regional Science Center for this opportunity, and I hope I can enlighten you about the this year Nobel Prize. And before going to this, I will just play with some numbers, and I feel that you will feel a great interest in that. So this is a, you can see this is a human cell, and this human cell is a very amazing thing. And I am going to touch upon very simple thing. One is the size of nucleus. Here you can see this is about 5 to 50 micron. And this can accommodate a huge length of DNA, which is approximately 1 meter. So you can understand this is a very small radius. And if you see the volume, this is approximately 5.24 into 10 to the power 5 micrometer square cube. And this, if you compare the length, then this is a, there is a 20,000 times difference. And such a big DNA can be accommodated in the cell. And this is not only the DNA, there is a lot of protein, as Bharadwaj very nicely explained you. So this is a very amazing thing. And now I will just take you to next, please. Yeah. So, so if you see the human body, so human body is approximately consists of 37 trillion cells. Here I will give you one very important fact that when we say human to any human or we name some human, then is it really human? Because you know, in class or in society, we call or we give the name to the majority, right? So I will just tell you an interesting fact about the human. So our cells, which is present in our body, is only 1/10. Most of these cells are microbes, which we call it as a microbiome. So we are only 10%, and rest of 90% is the microbe. So if I am calling Dr. Aditya, I don't know I'm calling to Aditya or to his microbe. So this is a one interesting fact which you will like it. So if we just hold on, can you go back? Yeah. So if we take out the all DNA and join it, then this will be a very big distance. It is about the 37 billion kilometers, which will make a uh, four, um, 4 times distance or 4 rounds from Sun to Neptune. So can you imagine these many big DNA we are holding? And one very interesting fact about the DNA is whatever DNA we have, only so far we were knowing that only 2% of DNA is making protein, as Dr. Bharadwaj explained you the importance of protein. So protein is the most important thing. This makes a structure, your nail. This makes an enzyme which catalyze all very, very complex biochemical reaction, which you can't imagine. Even the big chemists, they are trying to make some natural product or some all those things, which is very, very difficult under various conditions. So our cell or our proteins, all those things they are doing in a very simple temperature, pH, and all those things. So this is very amazing. I think you can understand. And our genome, which I have told you, only 2% is making a protein or coding. We call it as a coding sequence, which mainly makes a protein. And this protein can be used as enzyme, structure, and all those things, defense protein, and all those things. Rest 98% is a kind of black matter of the genome. Genome, we call the whole DNA in the cell, we call it as a genome, right? So 98%, why the nature made 98% genome, which we could not understand. So this work basically give us or put it some light over the 98% genome. I will give you one situation. For example, you are coming from home to school, okay? And if there is a no traffic light, or if the traffic light is not working, what will happen? It will be a chaotic situation. There is no control. Everybody will probably you have seen in India or in your Bhopal city. Sometimes the lights breaks, and then there it's a very difficult to reach to the destination. So similarly, this is happening in the cell. So protein, which is made by the cell, they are controlled by the protein. There are several factors which control the production of protein. So there is a something which we call it as a non-coding RNA. So non-coding RNA is made by this 98%, and this plays a very, very important role. It's a kind of traffic light, okay? Uh, this is basically controls whether the protein should be made or it should not be made, or there are so many things. When you will study the biology, then you will understand how this protein expression is regulated, okay? So this non-coding RNA, this work basically, so here I am just giving you another better picture. So this is only 2 to 3%, which makes a protein, and rest 95% is a, here you can see there are housekeeping genes, which basically plays a very important role, or it helps in making protein, okay? So another is, probably you might heard that there is a tRNA and ribosomal RNA. Probably you have studied in class, I think ninth or 10th or onwards. And there are so many regulatory RNA. These are this thing. So snRNA, SNO RNA, piRNA, circular RNA, microRNA, and long non-coding RNA. So the work which was discovered in this year, the Nobel Prize which they got, basically discovery of this microRNA. Okay, so I will give you a little quick overview. So this work was done by Dr. Victor Ambrose and Gray Ruken. So they basically use one animal model. We call it as a, so it's not getting- so there is a one uh, round worm kind of, it's a worm. Basically, this is a very microscopic worm, and this microscopic worm basically has a very defined thing. So now we know the genome of this worm. We call it as a Sinorhabditis elegans. Okay, so this worm has a few cells, and this worm basically used for the study of cell or cell-cell interaction or various complex biological process. Why it is used? Because this is a very simple compared to the human or mice. If you do study in human and mice or any animal,
इसका
it
भी
will be a very complicated. Okay, so they have basically find out there is some sequence of RNA, a very small sequence of RNA. Probably you know the difference between DNA and RNA. Okay, so there is some very small sequence of RNA which is about 18 to 22 bases. So these small pieces of RNA basically regulate. Do you know the how the genetic information flow? So there is a DNA. From DNA, there will be a synthesis of RNA that we call it as a transcription, right? Are you aware about this thing? And this transcription is followed by the protein synthesis. We call it as a technically translation. Okay. So, so there is a place when there is a formation of RNA to protein, we can also fine tune whether this protein is needed. If the cell needs this protein, then we can tune it, or we can degrade the RNA. In that way, we can reduce the protein expression, okay, or production of protein. So these microRNA is basically the post-transcriptional regulator, or you can call it as a fine tuner. Okay, I will tell you the importance of this discovery. So so they found out that this microRNA is highly conserved from C. elegans. Here you can see this is a picture where I can show you this is highly conserved in various in animal. It is present in human, mice, and different small organism, big organism, and all those things. So this discovery basically plays a very important role. Why? As you have seen in previous talk by Dr. Bharadwaj, that the protein is very important, and there is a something known as mutation. Okay, so if this mutation will happen, then protein will change. This is one outcome. Another outcome could be the protein production level can be also changed if there is some mutation, okay, or some genetic problem will happen. So this when this microRNA is discovered, then this can be also used in a therapeutic. So I will show you few examples by our study. I work with microRNA and our defense system. Okay, so these are few papers which basically showed the discovery of this microRNA, and this microRNA received a lot of. So now we know this microRNA is playing very important role, and we can basically control the all cellular activity by manipulating the microRNA. Okay, so this microRNA, now you know that the genome is same in all cells, right? You all agree? But some cell make a muscle cell, some cell make a brain cell, some cell make a liver cell. So all this thing, different thing, is basically governed by so many factor, and one of the very important factor is the microRNA. Okay, so microRNA is playing very important role in changing the expression or suppressing the expression and all those things, and by that we have a different kinds of cell and different kinds of situation or different diseases also. Okay, so here I am just playing one small video which will give you the overview about the microRNA.
Profiling microRNA by pathway and disease. MicroRNAs play an intricate role in gene regulation, and understanding their biogenesis and expression patterns is important in defining their biological functions. Biogenesis of microRNA begins in the nucleus. MicroRNA may be expressed in the same manner as any other gene and is often hundreds of bases in length. Before the primary microRNA transcript leaves the nucleus, it must undergo processing. First, Drosha removes the tails of the primary microRNA, leaving a shorter stem loop structure referred to as pre-microRNA. Pre-microRNA associates with an exportin 5 complex and is escorted from the nucleus into the cytoplasm. Once in the cytoplasm, the pre-microRNA is released from exportin 5 and faces further processing by the dicer complex. DICER removes the stem loop from the pre-microRNA, resulting in an asymmetrical, double-stranded RNA of 20 to 25 nucleotides. This double-stranded RNA associates with the RNA-induced silencing complex, also known as RISC. Once in the complex, the microRNA is unwound and guides RISC to conserved recognition sites in the target messenger RNA. Binding of RISC to the target triggers the silencing effect.
This is how the microRNA is generated into the cell, and I will show you another video which shows about the application of this microRNA.
Our body's ability to move starts in the brain.
So this
Upper
is
motor
talking about
neurons
one
send
disease
signals
known as
down the spinal
ALS.
cord to lower motor neurons and then to muscles. When muscles contract, we move. One type of ALS is caused by mutations that are inherited in a gene called SOD1. The genetic defect
mutation,
carries down to the messenger RNA, producing a mutant SOD1 protein that accumulates, harming the cell. Eventually, this harmful buildup takes away a patient's ability to move. A first-of-its-kind gene therapy clinical trial, led by the University of Massachusetts Medical School, aims to reverse that damage using something called a vector. In this instance, an adeno-associated virus is being used. AAV is a naturally occurring, harmless virus, but in the lab, gene therapy researchers transform it. They re-engineer parts of the AAV, replacing the guts of the virus's DNA with a synthetic microRNA that has been made specifically to target that defective SOD1 gene. It represents a new type of medicine that works on a genetic level inside of our bodies. This new recombinant AAV is then repackaged inside the harmless vector to be delivered to the study participant. In UMass Medical School's SOD1 clinical trial, the vector was injected into the spinal fluid of the trial participant, where it attached to the surface of the lower motor neuron, delivering a genetically modified therapeutic DNA. Now, with this therapeutic DNA inside the cell, it produces a synthetic tiny RNA that neutralizes the effects of that defective SOD1 gene. As a result of this gene therapy treatment, the cells produce far less of the harmful protein. Physician scientists hypothesize this could slow down or even reverse the progression of SOD1 ALS.
Okay, this is just an overview. I will just quickly tell about, yeah. So, so you know the influenza. So I think you all are aware, right? Influenza is, and every year there will be a change in influenza. Sometime you will hear that H1N1, H7N9. So basically the influenza viruses keep changing. And therefore, this is a one very big challenge to the biological or biomedical scientists to make a appropriate vaccine because they keep change their phase. Okay. So this is our laboratory work that we found out that there is a one microRNA. This here you can see microRNA 485. So this microRNA is produced in our body and basically it controls the antiviral factor. Okay. And this microRNA can also bind to the genome of influenza virus. And that genome is very much essential for the viral replication, making a copy of the virus. Okay. So basically, this microRNA targets the polymerase of influenza virus. Okay. So if we can target that the gene the polymerase which the virus cannot afford to change. If they will change, then virus will not exist, right? If they cannot make a copy and if they will make some change in that protein, then that will be not possible. The virus identity will be lost. So we discovered that there is a one microRNA 485. This can bind to that essential part. So these days you are also hearing that now there is a drug resistant microbes like tuberculosis. There is a extremely drug resistant Mycobacterium tuberculosis. There is a drug for the influenza virus, probably you might be aware that there is some drug which can be used for the treatment of influenza. But the influenza virus developed the resistance against those drug. Okay. So in that scenario, we need to change the therapeutic strategy. So far, whatever the treatment is there, it is basically a small molecule based treatment. Okay. And the microbes develop the resistance against those small molecules. So this could be a future drugs. This can be used as a future drug, but it is, I cannot say that this will be. The reason is that this microRNA can also regulate several genes. Okay. So before saying this thing, okay, this is a drug, I cannot say that this will be a drug, because there is so many side effects. There is a possible side effect. But this can be achieved. We can overcome those challenges by or with the help of chemistry. Okay. So we can design the basis in such a way that this will have a very high affinity to the influenza genome. And in that way, we can selectively treat the influenza. Okay. So this is one example. This is one of our not working. This is a one example where I am showing that there the virus which we think that it is a very simple microbe, but this is also doing lot of research. Here I am showing that there is a one microRNA. Here you can see microRNA 324. Okay. So this microRNA 324 is basically an antiviral. They block the influenza virus. But what influenza virus is doing, they are making changes in their genome so that the the blockage will be relieved. Okay. So here you can see they change the here you can see they So here you can see this, they change the basis. So microRNA basically interact with the transcript by simple Crick and Western pairing. Okay. So you know A pair with T, G pair with C. Okay. So these are the codes, right? So they changes the basis so that they can evade the host or the microRNA mediated defense. Okay. So this is another example. So again, if we make some therapeutically or stable molecule, then we can make this as a drug for the influenza virus. But there are a lot of things before saying this thing. This is another example. You know, when you have a fever, then you take a paracetamol or ibuprofen, right? So they are again the small molecule based drug which basically act on a one enzyme, very key enzyme, which we call it as a cyclooxygenase, okay, which makes a prostaglandin. Here we found out that there is some microRNA by which we can control all kind of inflammation. Okay. And this could be a very important for some autoimmune disease. You know, autoimmune disease. So our defense system start killing the our own cell, that we call it as an autoimmune disease. And this is very hard to treat. Okay. So we can use this kind of microRNA in order to control those disease. Okay. So this is all, and I will stop here, and thank you very much. If
Aye. You, uh, yes, sir. Thank you, Professor Kumar. Sir, that was really an eye-opening session, and I'm glad that this is a packed auditorium, and most of the students are really curious to understand and discuss later on when we will have this open forum. As we understood, when we do live streaming and other stuff, we always try to reach a larger number of audience across the state or across the nation or maybe beyond boundaries, because YouTube has global reach. But sometimes we always feel live streaming may not be very powerful media, because at the time when we are streaming some program may not be reaching to the people because that is the case in science. Science takes backseat. We need to take at front. For that matter, we have to make more popular, more create more awareness, more engaging and participatory sessions, so that the dilemma between, so-called technically and scientifically sound at area and fields and the growing area. So we need to bridge the gap. Definitely, it is a very great thing. ISA Bhopal CS2 department, with the great support from their team and Professor Kumar, Professor Satyamurthy, and Professor Sharma, they all have discussed beautifully about this year's Nobel Prize in very gist. And considering the time challenge, it is looking like international conference, and we have a very small time window to present our idea. But your audience is very different today, and I'm glad that it is a mix of class 9 to 12th and also engineering college students. So now let's open this forum. I'll ask one by one. Let's discuss. Then afterwards, we'll take a few questions from the audience, from schools and colleges also. At the same time, सर जैसे कि आपने बताया है कि माइक्रो आरएनए के बारे में आपका एक इम्पोर्टेंट इनिशिएटिव के बारे में आप एक्सप्लेन कर रहे हैं, जैसे फिजिक्स में हम बात कर रहे हैं आर्टिफिशियल न्यूरल नेटवर्क, और वो एएनएन जो है इतना इम्पोर्टेंट क्यों हो गया, और नोबेल प्राइज फिजिक्स में अगर लोग ये थोड़े से कंफ्यूज भी हैं लेके कि आर्टिफिशियल इंटेलिजेंस को एक बहुत बड़ा एक बल मिला है, ताकत मिली है इस तरीके के फाउंडेशनल फिनोमिनल वर्क डन बाय प्रोफेसर हॉफील्ड एंड प्रोफेसर विंटमैन, जो आज जाके शायद मॉडर्न एज में एआई या डीप लर्निंग और मशीन लर्निंग की बदौलत इतना एडवांस हो रहा है. आपका पर्सपेक्टिव जानेंगे शर्मा सर. एंड देन सर विथ यू आई वुड लाइक टू अंडरस्टैंड कि सत्यमूर्ति सर के साथ में कि आपने अभी अल्फा मॉडल का एक ओपन सोर्स वर्जन दिखाया कि बिफोर द नोबेल प्राइज दैट वाज नॉट ओपन आई थिंक. The latest version is now open.
Yeah.
So it is open in the public domain. Anybody who is interested in the field, they can play with that and they
Yeah.
can download, they can
That's
actually
right.
use. कैसे टेक्नोलॉजी, कैसे प्योर साइंस मिलके हमारी ग्रोथ को, एडवांसमेंट को एक पुश दे रही है. और आपने भी बहुत कमाल रूप में सर आपने जो बातें रखी, मैं आपसे भी जानना चाहूंगा कि ये जो पूरी डिस्कवरी है किस तरीके से कैंसर और डायबिटीज के क्षेत्र में ये पुश करेगा. तो लेट्स स्टार्ट विथ यू. Biological sciences is prevailing because we have observed very categorically in COVID period, it is the biggest challenge. So the way we are investing in other areas, we have to invest a lot in the field of biological sciences for making our civilization future sustainable. Then only future exploration will be a reality. So sir, over to you.
So the microRNA, the discovery of microRNA is a really very critical. The reason is that, as I told you, when there is some problem in a body, okay, so then there will be a change in expression of protein, as you have learned from Dr. Bharadwaj, that protein is a very important. But what is the behind? Like, so you have learned that AlphaFold 2, the behind science is artificial intelligence, which is explained by my colleague Dr. Aditya. So similarly, so when there is a protein and protein regulation, this protein regulation is not only completely sufficient. This is mediated by one nucleic acid or RNA based regulator or fine tuner, which we call it as a microRNA. So when the disease happen, the expression of these microRNA changes, and this will reflect in the blood, and this will if we learn those microRNA, we can diagnose, number one. Number two, if we learn more about these microRNA, we can change the course and then we can correct the disease also. So this is in very broader term I am telling you. This is not only for human, this is also true for plants and other living organism. Yeah.
And next question is for Professor Sutyamurthy. Thank you for wonderful. Please give round of applause so that the energy can be, restored in this auditorium. One more time, let's clap one more time with full energy and enthusiasm. Wow, thank you. I'll ask my team, come here, please, one more time for our whole audience. Yeah, so that was a small exercise also, so that this energy level can be restored once again with full attention. Professor Satyamurthy, we would like to know that as we understood, this year's Nobel Prize in Chemistry was a great combination of technological demonstration and also the very phenomenal work of protein. What I understood, the protein is very critical and important part of a human body, and amino acid is responsible for that also. So how you can relate and can you explain in very lucid manner for our school audience the protein and its connection with amino acid and the source of protein for vegetarian people? What I understood, I think, is a pulses only on a larger chunk,
Significantly, yes.
significantly,
Yeah,
a
yeah.
larger amount of whole
That's right.
protein source.
Yeah.
So can you relate these three, and you can also shower lights how this Nobel winning work will take lead in opening new areas where technology and science can grow hand in hand for future sustainability.
Sure. So to the first part, I'm not the best person to go on which foods will give, but as you rightly pointed out, pulses and dal varieties are extremely healthy towards acquiring necessary proteins. Not all amino acids are actually synthesized by the body. There are few essential amino acids which you have to eat to get. The other ones the cell can actually synthesize based on whatever resources it has. Okay, so definitely nutrition forms a very major part. Milk, for instance, has a lot of proteins, casein. So definitely making sure there is a balanced diet to ensure for growth of muscles, protein is extremely essential. Now, coming to the next aspect, as my colleague here nicely pointed out, I actually work in the field of nucleic acids while I've done my PhD in proteins. Proteins are kind of the workhorses in terms of getting their job done, but that's not without the presence of nucleic acids like tRNA, mRNA, and miRNA, and so many other things for the G quadruplexes that are being present. So, with that being said, where does the discovery, or rather the contribution of the Nobel Prize goes? I didn't have the time to say because I think I took extra time. Is that one could actually design proteins for some artificial purposes. There are design strategies going to figure out whether a protein can chew a plastic out. For instance, if one could design a protein that takes care of reducing pollution in very different forms, that will be extremely useful. And the example that I showed from David Baker's lab itself, where they were able to sense a small molecule, which was never the portfolio of a protein to do. They figured out, okay, this small molecule cannot be easily sensed in the environment. So they came up with a program to say, okay, if this is the fold, it can sense this small molecule. They gave the computer program, it came up with a sequence, they folded it, and experimentally they're able to show it can gain some artificial functions in it. So one can actually start making motors using proteins, which are very critical for many other functions. Actually make drug delivery systems, like he was trying to point out, the virus is actually a protein code. So in order to deliver a drug, there are ways of getting the drug through. You cannot just give the drug as it is. The small molecule or even the miRNA won't get in. You'll get disintegrated. So therefore, there are, as they nicely showed this nanoparticle made of the protein, protein design has a huge focus on purposes that are not available naturally. And if we can design things that will help us. Now coming to protein structure prediction, it has already contributed towards many diseases that have been not paid attention. I forget the actual term for these. Neglected diseases is the term. Yes. So many neglected diseases, science funding agencies do not give the money for. And what they were able to show with this AlphaFold 2 itself was to show the potency that if you have a sequence, I can actually give you an idea of structure that doesn't exist in the protein data bank. So there is a huge leap when it comes to what we stand to learn just from running a computer program. And once we get the structure of the protein, we enable ourselves towards targeting diseases that were previously neglected largely. So that's where the major purpose comes from with these techniques.
Oh, thank you. Please give a round of applause for wonderful, perspective on that area. And so from both of you, I want to understand the way we are growing in this field. Now, this mRNA or maybe alpha mole model based prediction and also the protein creation. Are we going in that direction in future with the help of technology and the growth of biological sciences in collaboration with chemistry? There's interdisciplinary field of sciences, finding out the problem inside the body. before actually it is going to be a very worse situation, especially in the case of cancer. We actually find very late there is a problem inside the body. So auto healing, or we can trigger ki at least tracing the disease, or maybe about to any situation can come up that can be, uh, traced very soon and for the proper medical treatment. Do you have any say on it?
Yeah, actually, this is our work. I'm working with the head and neck cancer, and I will not reveal the the original project, but we are developing something like that. We can know in very early stage if this project will be successful, even the cancer is not visible or just at cellular level. When there will be a changes, then we can find out whether the individual is going to have a cancer or not. So again, so when the full blown disease appears, from that time, or before, quite early before that time, there will be a changes in the cell, okay, and the cell will throw out molecules outside, which will be the signature of disease. And eventually when cell become when those kind of cell will be more in number, then that will be a kind of visible disease. So we are trying to figure out, to be very honest, that can we catch up those signature quite early. For example, so head and neck cancer is linked with the that nose, throat, and all this part, right? So if the individual who is chewing tobacco, for example, or the individual who is working in asbestos factory, so these individual are possible candidate for head and neck cancer, right? So if we have this system, we can screen those individual time to time, and if we see those signature, then we can recommend, okay, don't do further work in those kind of thing, or you go for deep medical checkup. So this is a kind of application of microRNA. And when we say, so now there is so many drugs which will be a next generation drug, that we call it as a MAPS. Probably you might heard, there is a monoclonal antibody which is basically used for the treatment of very complex disease. And this molecule is not the small molecule, it's a big protein. But making maps, maybe this will this the discovery which is which enables to design those proteins, probably this discovery will help. And in that way we can patch up both chemistry and biology, and we can help the humankind. Or this is not only for human, this is for all living system, that will include the plants, because the future problem will be the food security, okay. Yeah, yeah. So the food security is a going to be a very serious problem. So in order to overcome those things, this will help in all discipline of biological sciences, not only medicine, plant, and other environmental, and everything is linked. Try to understand. So in that way, this will be very big is baby step, I will say,
Okay.
for the future. Yeah.
Thank you. Thank you, sir, and for your perspective on it. And now, as we all know, this is the era of interdisciplinary sciences. So the bifurcation or isolation of individual areas or subjects are no more actually existing in reality at the forefront. This should also be taken seriously by our school audience. Categorization is not very good for the science journey. So the science is coming as a collective spirit. And on this note, I would ask Dr. Sarma, please, can you elaborate us how this technological breakthrough by Hoffield and Wintman is actually helping us in machine learning? And as we understood, mimicking brain, when this Nobel Prize was announced, so this word became very, buzzword across the globe. All media houses were talking about mimicking brain, mimicking brain. We all know brain is brain. We all have brain. how mimicking brain is actually working in this area, which we call by mapping neural network. So please help us to understand what is the neural network inside our brain and how we are actually connecting with artificial neural network, and then it is leading to mimicking a brain. Please, sir.
So there's a bunch of different ideas which have been explored here. So first, let's start with the idea of interdisciplinary sciences, right? I mean, so this year's Nobel, for example, in physics, has already created a bit of a controversy in terms of whether it's really physics that has happened here, and whether these gentlemen, although their contribution, nobody doubts that their contribution is wonderful, but whether it is really physics that went into it. And so my own answer to this would be, I would say that the ideas which go behind, the developing all this technology is definitely very much physics. I mean, so this goes back to the 70s and 80s when people were studying glass models, like window glass that you see. As you might know, it's to be thought of as a super cooled liquid. So what happens there is, I mean, it's neither a solid, not quite. It doesn't look like a liquid, does it? I mean, the window glass, it has properties which is similar to that of a solid. But really, what is happening is that it is stuck in one of these metastable states. It's a little bit like this. You drop this ball and it falls down to one of the valleys. And so the state that your window glass is in one of those kind of metastables. And if you wait for long enough, it's going to change to something else. So the time scales involved are very, very large. And so it was Hopfield's ability to these ideas from spin glasses. So the idea, the fact that there are so many metastable states, and whether it can be used as a model for associative memory, right? So there is a leap of imagination involved here. So ultimately, science moves by these kinds of creative insights that you pick from one field and apply it somewhere else, and then take from there and a third the place, and keep on doing this. And in the process, something which none of the scientists originally who came up with these ideas never imagined perhaps that this is where it would go to. Even Hopfield himself probably never thought that there would be a day when we would have this kind of a device. And I asked them to write a poem, and then the software is able to do it. Or things like the kind of amazing things that machine learning tools are able to do nowadays, right? So this aspect of interdisciplinary research is, you should just look for creative possibilities and break these barriers, and uh, come up with ways to merge ideas, no matter where it comes from. I mean, good ideas should be valued, and always on the lookout for using them in some completely different ideas, places. That's how science moves. That is point number one. So the other thing that you were asking was about where we can see these applications, how they may evolve in the future. It looks like AI is going to revolutionize pretty much every field that you can imagine. So we maybe 10 years down the line, an event like this, we might have robots sitting here and describing science to you. I mean, it's not such an outlandish possibility, right? I mean, so because ultimately it's already happening, right? Right. So even like so we can imagine scientific research being entirely commissioned by robots. After all, they are very smart, very efficient at taking like tons and tons of information can be used, right? I mean, but so you pretty much every field of human activity is going to be touched by AI. But hopefully, I am optimistic that it will be done in a good way, in the sense that I mean, there are all these doomsday predictors who think that a lot of bad things also will happen. But I'm hopeful that, I mean, every new technology that humankind has come up with, initially there would be all these kinds of noises which said that, oh, terrible things will happen. But we have figured out ways to counter that, right? So hopefully this revolution too will go in the right direction, positive direction.
Thank you. Thank you. Please, for the Dr. Sharma. Considering the time limit, on the last note, I wanted to tell, but now I will redefine my own statement that idea is very critical part of any journey. Sir was talking bad or good idea. Idea is always an inspiration for evolving for better and better, a better version of its own idea. So let's hope with this positive attitude that interdisciplinary field of sciences, these years Nobel Prizes are just a catalyst for us to rethink about on these few areas, but there are many areas in the field of sciences, including mathematics. They are actually growing and evolving and revolutionizing the field also. They may not be a part of Nobel winning work, but they are actually equally or maybe most important sometimes beyond the Nobel winning work also, because it Nobel Prize may not be given to everyone. So the work is done by lots of people across the world. Let's celebrate. Let's give round of applause for all the scientists, our Indian scientists, all the labs, research organization, and also school and colleges. They are the future. Actually, they can take a part of it. And that's the last. Yeah. So on last but not the least, very critical part, definitely that is the Q&A session. I understand some schools are having a commitment of returning on time, but time is very universal quantity and challenging also. So flow of time is for all of us a challenge. So let's embrace the challenge, and this we open this floor for taking few questions. So if you have any question, please feel free to ask and interact with our today's panelist. Yes, there is a question. ये माइक उनको दे क्या माइक उनको कोई दे दो दौड़ के इसी में बोलेंगे तो सब
Good afternoon, all of you. My name is Akshat Verma, and I am from CS department. And my question for Sharma sir, sir, what is the speed of neurons?
Speed of neurons which are in the brain, or
Yes,
this
yes, sir,
artificial?
brain.
I don't know. I mean, so we can always come up with some estimates about, I mean, in terms of uh, how quickly you recall and stuff. I mean, it would be in the order of probably a few milliseconds, I would guess, but I'm not sure. I mean, I don't know. So, but even faster. Faster, yes. So, but what is the thought process behind this question? Maybe that is more
No,
interesting than the
sir,
number.
you have told in the presentation that aloo paratha and that. So,
No, that, yeah, see, that is, see, that's an instantaneous memory, right? I mean, the fact, so the all that I wanted to say was that if you had this kind of a linear memory, right? I mean, if there were actually a register sitting inside the head, and if your brain had to go and scan, okay, is this does this correspond to aloo paratha, this second one, third one, all the way up, if it were to go in a linear order, it would take probably like the age of the universe, the time scale involved. But it's not taking you that kind of time, right? I mean, you're immediately able to not only say that it is, you are connecting it to aloo paratha, but there are many other memories associated with it, like some red color, or I mean, I don't know, like the place where you were. All of these things are coming. Why is that happening? That's because the algorithm involved is a little bit like the algorithm which Hopfield came up with, which is basically, it's a kind of a descent into energy space. So what you can associate an energy, and you say you flip a certain spin or a certain neuron, and then you go to the next one, and then you keep on flipping it if it is going to go downhill in energy, and very quickly you will go to one of the minima. You may not go to the absolute minimum, but that doesn't matter. So that is where some memory is hidden, and so immediately you are able to connect it to that particular memory, like the J that we saw,
yes, sir.
right? So yeah, so the time scales, the precise value of the times involved are not as important as realizing that it's not some astronomical times, it's instantaneous, right? Okay, thank you.
Sir, another question. Uh, we can control neurons, sir.
Of course, you're controlling right now while you speak.
That's right. I mean, so they're getting spontaneously fired. So there is something called the Hebs rule,
No,
which is
that speed of neurons we can control. That
not
controls the
consciously,
mind.
not consciously. It's already happening inside.
Okay. Thank you, sir.
Sure. Okay. When did you want?
Uh, my warmest greetings to all the professors and teachers here. I am Maisha Khan from class ninth from Carmel Convent School, and my question is to Sir Sharma. Sharma sir, as you talked about memory, during your presentation, I wanted to ask that what is the concept and thought process behind deja vu? Usually, whenever we have experienced some sort of situation and we have this sudden realization that we have experienced this somewhere else, but yet again, we cannot put our finger on it. So, can you please explain it to me? It's been a really interesting.
Difficult question, but in the sense that I mean it's quite away from my expertise. But the thing is that the only point I'm trying to make here is that there is this notion of a memory. So deja would be some special kind of a memory, perhaps, right? I mean that is, I mean it's the neuronal network that is actually inside the brain, right? So is actually way more complicated than the kind of toy models that we are playing with, right? I mean, so in fact, I was reading somewhere that each individual neuron, which we are modeling as simply as something which can be either up or down, 0 or 1, itself should be thought of as a full network, right? I mean, the human body, the actual brain that we have is like enormously more powerful than these devices that we're toys that we are playing with. But even these toys are already giving us so much, variety of possibilities have opened up. The reason why these things have become so exciting in the last 10 years or so is because of the computational power that we now have. I mean, these models have been around for like 50, 60 years already, but now they're making a huge impact because we have these fancy computers which have come in. Now, if the models themselves become, if there is something like deja vu, perhaps there's a deeper biological, neurological aspect itself. I don't know how well that has been studied. If those kinds of fancy things also can be incorporated into an artificial neural network, imagine the possibilities for the future.
Yes, sir. Thank you so much, sir.
Yeah, just yeah. Thank. So just I want to say that the artificial intelligence basically worked on the all information. It's a learning and quick responding, right? It's not that this will create a new based on the existing information. Am I right? Yeah. So so once we will understand this deja vu, then we can use the artificial intelligence, and then we can do it. Yeah.
थैंक यू शर्मा जी और थैंक यू कुमार साहब। मैं ये कहना चाहूंगा कि सवाल जिस तरीके से ट्रिगर हो रहे हैं, इट इज वेरी इंटरेस्टिंग एंड वेरी इंस्पायरिंग फॉर ऑल द पैनलिस्ट एंड ऑल ऑफ अस, बिकॉज दैट इज द मेन पार्ट ऑफ साइंस कम्युनिकेशन। कार्ल सेगन जो है बहुत फेमस एक साइंटिस्ट हैं, हुए हैं, जो एक रोल मॉडल भी रहे हैं साइंस कम्युनिकेशन की दुनिया में ग्लोबली। उनने बहुत फेमस लाइन कही, उन्हें इंटरव्यू अभी हाल में हम देख भी रहे थे दोबारा, कि वो किंडरगार्टन में हम जाते हैं तो बहुत सारे सवाल आते हैं, बच्चे इतने सवाल पूछते हैं कि टीचर्स को कंट्रोल करना पड़ता है, और जैसे ही हम हायर क्लासेस में जाते हैं, अगर ग्रेड 12 में या क्लास 12 में पहुंचते हैं, सवाल नदारत से होते हैं। तो हमारे सवाल खो कहाँ जाते हैं रास्ते में, उनको वापस हमें रिवाइव करना है। द कल्चर ऑफ क्वेश्चनिंग इज वेरी क्रिटिकल एंड वेरी इंपॉर्टेंट फॉर द फील्ड ऑफ साइंसेज। एक्चुअली आप सोचना बहुत जरूरी है, और मैं टीचर से कहूंगा अगर बसेस लेट भी हो रही हैं तो मैम प्लीज विथ योर मैनेजमेंट ट्राई टू मेक श्योर कि ये जो गोल्डन अपॉर्चुनिटी है इंटरेक्शन की, वो मिस ना हो। वी विल अलाउ द स्टूडेंट ऑन द लास्ट। यस, और यहाँ पर भी है। यस प्लीज उनको माइक दे दीजिए।
Just out here, just I would like to add a point about ISER Bhopal and CS2. For the teachers here, please note that ISER Bhopal conducts regular workshop for school students, and we keep inviting students to come over summer and spend a week with us in the hostel and all that. So please have an eye out for what all kind of workshops we call summer, winter. So please look out, and as he rightly pointed out, questioning and discussing with students matter a lot, and you'll get more opportunity when you're at ISER Bhopal. Question, please.
Hello, I'm Vedant, and my question is to the sir wearing the blue shirt. You
Blue
take
shirt.
the mic.
Right. Yeah.
So the question is, how does the structure of proteins help in predicting any, drugs against viruses? Help in making drugs against viruses?
Thanks for your question. The question is, if I may repeat, proteins targeted for viral diseases. Is that what your question is?
My question is, how does the structure, how does knowing this, guessing the structure of proteins help in making any drugs or drugs that would fight against viruses?
Excellent.
I'm Vedant and I'm from 8th.
8th.
With school IPS.
Okay. Thanks for your question, Vedant. So the thing is, generally the way drugs work is that proteins generally have a certain structure, and the function happens in a certain place. Think of a case where you have a lock and a key gets in, right? And the example that he was trying to give, I can actually use it. For instance, you have a certain RNA that codes for a protein, and that causes a disease in the example that he showed. And what does the miRNA do? Go and stop the place where the protein gets made. You can imagine the case like you have a car and you want to start the car with a key. But in the keyhole, let's say I put some other key and occupy it, which cannot start the car. Do you realize that is how a drug works? Yeah, you have a key which goes into the keyhole of the car, but will not start the car, but will jam the keyhole and will never get to start the car. So that's how drugs work. Definitely for that, understanding how the keyhole looks also matters. Okay, you cannot just put any key which will not get into it. So understanding a structure of a protein and therefore then figuring out where to target it, meaning that which keyhole to put. That might be multiple. If you take an aircraft, for instance, there'll be multiple keys to put in, right? So one has to figure out what is the most effective place to put such that the aircraft won't take off, for instance, right? So that's how proteins also work. Proteins have something called an active site, and that active site gets modeled, and then people start doing other computational techniques called docking with different small molecules and figure out, okay, could this be a potential drug that could work against a certain disease? And for instance, people have been trying to do that for different viral ones, but viral ones are very tricky. As he rightly pointed out, the mutations happen at a much faster rate, and it's not very easy to target viral proteins with small drugs. Although vaccines are possible to understand how they end up coming and reducing their impact, having direct drugs against viruses is not a very easy possibility. But there are other ways to tackle it. Maybe he'll add more to it.
So viruses has so many ways to escape. One is that they will keep changing their face, like influenza virus. So they are they are expert in that. Every year there will be a different kind of influenza virus. I'm just giving a one example. Some viruses use our defense system in order to infect and make it more severe. The best example which is running these days is the dengue virus. So I think in the city, the dengue virus is picking up. So they use our defense system and then they evade the immunity, our defense system, and then they cause a very severe disease. So the protein structure is a very important. So everything is try to understand. In living system, most of things are protein and modified protein, which we call it as a post translationally modified. It's they are decorated with some sugar molecules, some small lipid molecules, and all those things. So if we know the structure, then we can design a molecule which Dr. Bharadwaj told as a key. We can design a molecule which will be a fit key, and then we can screen which key is the best in order to check the viral infection. So this is the importance of discovery of protein structure. And previously the protein structure was done previously also, but it is a very difficult to purify the protein and then crystallize and then subject it to the X-ray diffraction, and then you find out the structure. Now this is something like, to be very honest, it is something like a chat GPT. You say that, okay, this is the amino acid sequence, can you tell the structure? So this AlphaFold or AlphaFold 2, which is released yesterday, this will do the job like chat GPT. Okay.
Next question.
Yeah, considering the time challenge, can I involve teachers? Can we take one or more questions, one or two questions with your kind permission? I think they are running out of time already. Their buses are waiting. So, ma'am, you have to tell us because they are very curious. It's like a nuclear reaction now is happening. So, can we take one or more one questions? Yeah, then I'll give. Yeah, please. Kamala Nehru School.
Sir, I am Ankit Ghimire from Kamla Nehru School, studying in 12th standard. Sir, my question is that universe is infinite or it is limited? Because we have images like observable universe, which looks similar to our neurons. So, what can be the possible reasons for it?
Yeah, so it's not possible to answer a question like this within the realm of science, right? I mean, so you only have control over the observable universe. So you have information only about the observable universe. So if you ask a question, what lies outside of what you can observe, then you have entered into some realm of. Yeah, so I don't know. It's a good question, but I don't think there's an answer. I mean, you should keep it in your list of questions for you to ponder over.
On this wonderful note, we are going to culminate this program today. And with your kind permission, let's give a round of applause for our today's speaker, Opishani. And also, we are very much thankful to IISER Bhopal. Once again, Professor Govardhan Das, Director of IISER Bhopal, the whole CS2 team. Tongue twister, because they have a very big network of, scientists, professor, expert. They are working collectively for a very greater cause. The way sir has just explained about their programs also. So we should also be thankful to him for actually informing you about the ISIS activities. And on this note, on behalf of Regional Science Center Bhopal and entire family of our science center, all the schools, their management system, students, and our council, National Council of Science Museums, which is a scientific body under the Ministry of Culture, Government of India. Thank you one and all for giving your time. And now we are closing. Thank you. Keep learning and exploring the world of science. Thank you.